Hi everyone, welcome to Unit 3, the last unit of Biology 1. Today we're going to learn about the cell cycle or the life cycle of the cell. As always, if you click on this book, then you will get a copy of the guided notes, which you can later turn in for extra credit. Now, the cell cycle is the life cycle of the cell, and the cell is going to spend the majority of its life in something called interphase. Interphase includes G1, S, and G2 phases. So the entire life of the cell is spent in interphase, except for cell division, which we call the M phase. M phase includes mitosis and cytokinesis, which can somewhat happen at the same time. The goal of N phase is to split in half into two separate identical daughter cells. Now let's talk about interphase. I mentioned earlier that interphase is the entire life of the cell, but it starts out with G1. G1 is normal cell growth. It's actually called GAP1 phase, but I like to refer to it as growth 1 phase because I find it's easier to remember. Now, here we are going to have normal cell growth, and this can last the longest of all the different phases. In fact, it can last indefinitely. If the cell does not pass the checkpoint to go on to DNA replication and cell division, then we call it G0 phase, where the cell will stay in G1 phase almost indefinitely. Think about your spinal cord. Your spinal cord doesn't generally divide or grow, so it's considered to be in G0. But all your other somatic cells or body cells are in G1 phase. Here we will have two copies of the DNA, and we will be doing whatever job is intended for this type of tissue. For instance, if it's a pancreatic cell, it will produce insulin. If it is a stomach lining cell, it may produce stomach acid. All of this is happening in G1 phase. If the cell moves on to S phase, this is known as synthesis phase. What are we synthesizing? Well, DNA. During S phase, we will make a copy of all the DNA inside of the cell. So we actually end up with 4 copies of the DNA. Now, G2 phase, we have twice the amount of DNA that we need. Now in G2 phase, we are going to have twice the amount of cytoplasm, mitochondria, and other organelles. This is growth in preparation for cell division. So we're going to get bigger and ensure that the cell has enough stuff that it won't die if it divides in half. The next phase is M phase, which is cell division. Now, how do we go from one phase to the next? Well, we have to pass a checkpoint, and there is a checkpoint at each phase. For instance, if the cell does not pass the checkpoint between G1 and S phase, then it will stay in G0 phase. If it does not pass the checkpoint between S phase and G2 phase, then the cell will not divide, and it may even cause apoptosis. If the cell does not pass G2 phase, then it will not divide into 2 daughter cells, and it will just kind of stick around. This is really great because it ensures that no mistakes are made or no extra cells are made when we don't need them, especially considering density-dependent inhibition, meaning the cell will not release growth factors which are necessary for cell division or protein kinases that are necessary to pass the checkpoint if the cell feels crowded. That means we don't need any more of that cell because we have plenty of that type of cell. So the density-dependent cells inhibition will not undergo cell division, whereas if we release growth factor, the cell will divide. Now, what happens when a cell doesn't properly respond to growth factors or inhibition factors? Well, then we can form a tumor. This is when a cell stops responding normally to checkpoints, growth factors, or inhibition. It also is resistant to apoptosis and is very difficult for our immune system to target these cancerous cells, which can spread. Cancerous tissue generally is very resistant to density-dependent inhibition, and it also doesn't generally anchor to cells around it. And it's difficult for our immune system to target because these cancer cells are made from our own cells. So they look like our own cells. So any treatment that we have for cancer is usually targeted at cells that divide very rapidly because cancerous cells divide very rapidly. Now, let's go back to the cell cycle. We talked about growth 1 and growth 2 phase. Let's talk about S phase. S phase, or DNA replication, is going to happen. We're going to go from having 2 copies of the DNA to having 4 copies of the DNA. We're going to double all the DNA that's inside of our cell. This happens in a DNA replication fork where one strand is pulled to this side and one strand is pulled to that side. These original strands are now known as the template strands, and an enzyme is going to come in and match the base pairs to each of these template strands, creating a complementary brand new strand. This end result is called semi-conservative, meaning we conserved one strand and we have one new strand. So our template strand is old and our new strand is the complementary strand that was formed by our enzymes. So one daughter cell is going to have exactly half of its DNA be complementary and half of its DNA be template. This is semi-conservative. Now, this process is a little bit more complicated than I initially led you to believe. This is happening in this replication fork due to all these different enzymes. And it's all going to happen in one direction, the direction of the helicase. So in this image, the first step is going to be topoisomerase. This is an enzyme which its entire job is to unwind or uncoil the DNA that is oriented in a helix shape. So DNA is not naturally twisted. This topoisomerase will flatten it out. That allows helicase to bind, and helicase is going to unzip the DNA. If you recall from our earlier unit where we learned about macromolecules, these nucleic bases, these base pairs, are connected by very weak bonds called hydrogen bonds. So helicase doesn't have to expend very much energy to kind of just break apart and unzip all of those bonds. Now, we do have little proteins that are responsible for holding open the replication fork, but the next big enzyme is primase. Primase will actually add an RNA primer. So instead of thymine, it would be uracil. This RNA primer is only temporary though. The RNA primer gives DNA polymerase something to grab onto. And so the polymerase will grab onto that primer and then start replicating the DNA by matching and pulling in matching base pairs. Now notice that one strand is called the leading strand and one strand is called the lagging strand. That's because this DNA polymerase is moving in the same direction as helicase. So it can start at the beginning and then just kind of continuously go forward until the very end because it's moving in the same direction as helicase. In this case, we only needed one primer, and then we can just continuously match base pairs and replicate the DNA. But this DNA polymerase, it's moving in the same direction as this one, but it seems to be opposite. It's going backwards. That's because DNA polymerase can only add base pairs in a certain direction, in the 5' to 3' direction. Now, this image doesn't have the 5' and 3' labeled, but I will show you that in a second. Just keep in mind that since this polymerase is moving in the opposite direction as helicase, it can only move as far as helicase has opened. So, for instance, we're going to start here with an RNA primer, and we're going to move this direction, then release and rejoin to the next primer and move this direction again, and then release and rejoin with this primer and move this way again. This causes the DNA to be replicated in fragments known as Okazaki fragments. So it's fragmented and it takes a lot longer than the leading strand, which is why we call it the lagging strand. But don't worry, we're going to repair those fragments with something called exonuclease, which will remove the primer and DNA ligase will bind or repair all of those breaks. We also have other enzymes that kind of double check the work of polymerase. So here you can see the 3' and 5' ends being labeled. This has to do with the ribose, or the sugar that makes up the DNA backbone. Now, they're always oriented opposite of each other. So 5' to 3' is going to be one strand, and then 3' to 5' is going to be the other strand. But keep in mind that primase can only add base pairs in the 5' to 3' direction, such as seen here in this continuous leading strand. However, when primase moves in the 5' to 3' direction on the lagging strand, it has to do so in fragments. So it's actually replicating this strand, then releasing, replicating this strand, then releasing, replicating this strand, and releasing, causing those Okazaki fragments. Now, what I like to do is have my students write with a partner. If they have a blank piece of paper, they can draw a replication fork, and one student will be able to write continuously in the same direction as helicase, which in this case is going to the left. Miss Alsaba's class is my favorite subject. This student did not have to pause at all. They were able to write directly in the same direction as helicase. But this other student, who is on the lagging strand, had to write in fragments. So even though we're writing in the right to left direction, the same as this arrow, this student had to write science, love, me, makes, she, because these words go in this direction. These letters represent Okazaki fragments. So this is going to be the lagging strand, and this is going to be the leading strand. Remember, polymerase always adds base pairs in the 5' to 3' direction. So the leading strand is going to be working in the same direction as helicase, and the lagging strand is going to be working in the opposite direction as helicase. So I asked my students a couple questions, such as what happens in the G2 phase. This represents interphase, and interphase is separated into G1, S, and G2. G2 is known as the second growth phase, where we are preparing for cell division. So this cell is bulking up. It's going to gain twice as much cytoplasm. It's going to grow twice as many mitochondria. It's doing a lot of work in preparation for cell division. What stage does DNA replication happen? Well, S phase, which represents synthesis. And the section labeled I is known as interphase. Let's learn about the section that's not interphase, M phase, or mitosis. The M phase is when cell division happens. Mitosis is mainly focusing on the genetic material. Remember, in S phase, we duplicated how much genetic material we had. We went from having 2 copies to 4 copies. And in mitosis, we're going to be splitting that apart, going from 4 copies back down to 2 copies into 2 separate identical daughter cells. Cytokinesis, we can break that word apart. Cyto kind of reminds me of cytoplasm, and kinesis is the movement, such as telekinesis is moving something with your mind. Cytokinesis is moving apart the cytoplasm. So mitosis is the division of the nucleus, cytokinesis is the division of the cytoplasm, which includes all the organelles inside of the cytoplasm. This cytokinesis and mitosis is happening concurrently at the same time. Cytokinesis is really controlled by these microtubules, which will stretch and then pinch the cell in half, leading to 2 identical daughter cells, which has roughly half of the cytoplasm in each side. And this is controlled by the centromeres. Now, mitosis happens in 4 distinct stages: prophase, metaphase, anaphase, and telophase. This is where the genetic material is organized, split in half, and then turned back into 2 separate nuclei with exactly half identical DNA on one side and half the identical DNA on the other side. And we will have 2 copies of this DNA. So let's review prophase. Prophase is the first phase of mitosis, and this is where the nucleolus is going to disappear. The chromatin, which is just the jumbled up DNA inside of the cell that you can see right here, it actually starts to like condense and form into distinct chromosomes. So we start to see the chromatin form into distinct chromosomes, and then the centrosomes will separate and go to either side, one on one pole and the other on the other pole, and they will start to form these mitotic spindles or these spindle fibers. Now, when we are going through prophase, we are packing the DNA into these tight packages called chromosomes. So chromatin is naturally just kind of loose, jumbled up DNA. It's really easy to access. Enzymes have to access this DNA all the time, maybe to make a certain protein, access a certain gene, or even for DNA replication. Chromosomes are too tightly packed for any enzyme to get in and do anything, but it's really great for moving the DNA from one cell to another. So it's kind of like chromatin is when I have all my clothes just kind of laying on the floor or jumbled up in the closet. I can access my clothes. I can grab my shirt if I need my shirt. I can grab my pants if I need my pants. But if I want to move or travel, I want to package my clothes very tightly so that they fit in my suitcase. So this would be more like having chromosomes. And I can easily identify I have all of my black shirts over here and I have all of my red shirts right here. Everything is very easily identified and moved, but not so easy for me to access and grab onto. The next, here we see prometaphase, where we see the nuclear envelope further dissolving, and we can see the chromosomes have already formed, but now the spindle fibers are attaching to the connectocores at the centromere, at the center of the chromosome. The microtubules are going to attach to that connectocore, and they are also stretching the cell apart. Now, this is because one centriole is going to pull one side of the DNA, and then the other centriole is going to pull the other side of the DNA in kind of a tug-of-war situation. And this is happening at the connectocore. The next phase is called metaphase. Metaphase for me is easy to remember because M stands for middle. It is very distinct. You're going to see the chromosomes exactly lined up down the equator or the metaphase plate. Some people call it the equatorial plate, some people call it the metaphasic plate, but they're going to be lined up right down the middle. And this phase seems to stall. It seems to last the longest of all of the mitosis phases. Because these centrioles are pulling and tugging on either side of this DNA, trying to pull it apart. Next is both sides have kind of won their tug of war game, and they are pulling half of the DNA to one pole and half of the DNA to the other pole. And here we really start to notice cytokinesis because we see that stretching and elongation of the cell. We can also see that there's kind of nothing in the middle. We've pushed all of the organelles to one side or the other. So very quickly, we're going to run into telophase, where we further have separation of the cytoplasm, and now everything happens opposite of prophase. So in prophase, we saw the chromatin fold and condense into chromosomes. Now we see chromosomes unfold back into chromatin. In prophase, we saw the nuclear envelope dissolve. So in telophase, we're seeing the nuclear envelope reforming, and we have two of them now. And the nucleolus will reappear, and the cell will continue to elongate and even pinch into something called a cleavage furrow. As it continues to pinch, this is known as cytokinesis. And telophase and cytokinesis are going to finish up kind of at the same time, and we will end up with two identical or genetically identical daughter cells. Here it is all together. And like I said, we end up with two identical daughter cells back in interphase. So we start out at an interphase with one cell, two copies of the DNA. We're going to replicate that DNA in S phase, and we have 4 copies of the DNA here. We're going to separate and pinch apart, and we're going to end up back at interphase with 2 copies of the DNA. Now, this is in animal cells. Animal cells happen a little bit differently than plant cells because plant cells do not have these little centrioles right here. What they actually do is very similar. They have prophase, metaphase, anaphase, and telophase, but they don't have pinching in the middle. Remember that plant cells are surrounded by a strong cell wall. So what plant cells actually do, instead of pinching in half, they grow a cell plate or a growth plate in the middle from vesicles that are filled with the same thing that the cell wall is made out of, cellulose. So they do not go through cytokinesis. Instead, they produce a cell plate, which eventually will become a cell wall. And then each of these daughter cells will elongate back to their normal size. This appears similar to binary fission, which is what bacteria does. It elongates and then pinches in half. But remember, bacteria do not have a nucleus, so they don't undergo mitosis the way that plants and animals do. Now, looking at this image, we see the interphase, prophase, metaphase, anaphase, early telophase, and then late telophase. This question is asking us in which stage do we see nuclear envelopes reform and the chromosomes unfold back into chromatin? Well, that's going to be in telophase. In this image, it's asking us which stage most closely represents anaphase. This can be a difficult decision because these stages are not distinct. They're continuous, so they kind of flow one into another. Here I can very distinctly identify metaphase because I can distinctly see the chromosomes lined up on that metaphasic or equatorial plate right down the middle. Here, I would maybe call this early anaphase because I can start to see that tug-of-war game being pulled apart. And here I would definitely distinctly call this one anaphase. I wouldn't really consider this one anaphase because those chromosomes are starting to unravel back into chromatin. It looks a little bit more blobby. It's not as distinct as here. So I would call this early telophase, late telophase, and cytokinesis. Here, let's view a video of this happening in real time. There is no sound on this video, so you're not missing out on anything. But we're going to observe this actually happening in real time. Here we can see the cell. It has already gone through prophase, and I would call this metaphase. The chromosomes are distinctly lined up in the middle of the cell, and it's hard to tell, but they are doubles of each other. And in a moment, you'll see them very quickly split apart as anaphase begins here. Anaphase, and very quickly we will see telophase as that cleavage furrow forms right down the middle, and the chromosomes unfold back into chromatin. Here we can see this happening with multiple cells at the same time. It's stained a little differently. And here we'll view this last one, which I really like this video because metaphase is so very obvious. You can almost see the cell fighting over the DNA, being pulled to either pole. So here we see the DNA being lined up right down the middle. And metaphase kind of lasts the longest because it has to play that tug-of-war game. But as soon as anaphase begins, everything happens very quickly. We see anaphase, and then that cleavage furrow is forming, splitting the cell in two, and telophase continues, uncondensing that DNA. All right, everything that you just learned is mitosis. This is normal cell division in somatic cells, which is body cells. But what about the testes and ovaries? How do we make offspring? So now we're going to learn about meiosis. Now I don't have any tricks or tips on how to distinctly remember mitosis versus meiosis, except you just got to memorize the difference. There are some questions on the exam which will ask you to compare meiosis to mitosis. So let's talk about offspring. Now, a lot of animals will undergo asexual reproduction. Asexual reproduction is a great strategy because you don't have to expend a lot of energy trying to find a mate. As long as conditions are good and you have enough material and resources, you can just clone yourself. But the problem with asexual reproduction is that you get clones. There is no genetic variation, which means the population is very susceptible to disease, or if we're talking about bacteria, maybe antibiotics. So animals have, and even some bacteria have formed a way to undergo sexual reproduction, where we kind of mix up the DNA and make genetically unique offspring, which means the whole population has a better chance of survival, because if one individual is susceptible to a disease, that does not mean that the rest of the individuals are also susceptible to that disease. So sexual reproduction is a really great evolutionary advantage to the population. Now, we also have to talk about haploid versus diploid. I mentioned when I talked about mitosis that all of your somatic cells have 2 copies of DNA. We call this diploid, or 2N. In humans, we have 46 chromosomes, or 23 pairs of chromosomes. So you remember that distinct X shape. That's because we have 2 chromosomes that are homologous. They are related to each other. So for instance, the gene that controls hair color will be close to the gene that also controls hair color. We have 2 copies of this. These are homologous chromosomes. Usually we get 1 from our father and 1 from our mother. That is why we have diploid genes. But in meiosis, we're going to produce haploid cells, meaning they do not have a homologous pair. They are individual, 1 single copy of the DNA. This allows our gametes, or sex cells, to have more genetic variation because, like I mentioned earlier, we're going to give 1 from the paternal parent and 1 from the maternal parent. So we have 1 from the sperm and 1 from the ovum, or the egg. That's what we're going to be producing in meiosis. We're going to be producing a gamete. Now, meiosis. Meiosis is very similar to mitosis. It has prophase, metaphase, anaphase, and telophase. And we're going to copy the DNA so that we have 4 copies of the DNA before we get started. But we go through meiosis 1 and end up with 2 daughter cells that are not genetically identical. And then we go through meiosis 2, ending up with 4 daughter cells that are genetically unique, that have half the amount of DNA, or haploid, only 1 copy of the DNA. This means that in interphase, we still have 2 copies of the DNA, or 46 chromosomes. By the time we start prophase 1, we have 4 copies because we replicated the DNA in the S phase, or in the synthesis phase. We have 92 copies of the 92 chromosomes, not 92 copies, 92 chromosomes, 4 copies. We call this a tetrad. Then after meiosis 1, we end up with 2 copies again, 46 chromosomes, but they're genetically unique, unlike mitosis, which will be genetic clones. Then we go through meiosis 2, dividing a second time, so we go from 46 to 23. And this is called a haploid gamete. And this is happening in the testes and the ovaries. We'll discuss the difference between testes and ovaries at the end of the lesson. Here we can see everything all together. Here we have prophase 1, metaphase 1, anaphase 1, and telophase 1. We end up with 2 genetically unique daughter cells. Then we go through prophase 2, metaphase 2, anaphase 2, and telophase 2, ending up with 4 genetically unique haploid gametes. Now, since I have a tetrad, or 4 copies of the DNA, we have maternal DNA and paternal DNA sitting right next to each other. This is an opportunity for crossing over. The next opportunity for genetic variation is in metaphase 1, where they randomly align on the center, or the metaphasic plate. So we're going to talk about prophase 1 and metaphase 1. Prophase 2, metaphase 2, anaphase 2, telophase 2 all happen very normally, except that we're dealing with half the amount of DNA. So in prophase 1, we have something called crossing over. This is a great opportunity for genetic variation because, like I said, we have maternal DNA and paternal DNA. And since they're lined up close to each other in this tetrad, in this group of 4, if they accidentally touch each other, they can accidentally switch places. This is called crossing over. And it's fine if this happens because, as I mentioned earlier, the gene that is going to control eye color will be lined up right next to the gene that controls eye color. And the gene that controls hair color will be lined up right next to the gene that controls hair color. This leads to very genetically unique gametes. We can have a mix of maternal and paternal DNA in one gamete. This is kind of like if I have 2 cars, a red car and a blue car. Now, I have doors, windows, and wheels, and all of them are kind of the same, except that one is blue and one is red. So during prophase 1, and only during prophase 1, these things are lined up so close to each other that they might just switch places. My car is still functional because it still has wheels, but it just switched wheels. So it's fine if this happens. In fact, it's encouraged, or it's a good thing, because it creates more variation. The next thing that could happen that could possibly cause more genetic variation is called independent assortment or random assortment. Both independent and random assortment mean the same thing. It means that during metaphase 1, and only during metaphase 1, this does not happen during metaphase 2, only during metaphase 2, these tetrads are randomly aligned down the middle, meaning the maternal genes may end up on the left pole or the right pole randomly. This, combined with crossing over, can create an incredible amount of genetic variation. So for instance, in this possibility 1, we got all of the paternal genes on one side, so we ended up with 2 gametes that were exactly like your grandfather and 2 gametes that were exactly like your grandmother. This actually happened in my family because I have brown hair and brown eyes, but my son has blue eyes and red hair. This is a trait that my grandfather has. So this can happen. Another possibility that could happen is that they could not line up perfectly with grandmother on one side and grandfather on the other side. They could kind of randomly align, or independent assortment, where we end up with a mix and the gametes have some maternal DNA and some paternal DNA. This actually happened with my other son, who has hazel eyes and blonde hair. So he does not have the same genes as my grandfather, except that he had some of those recessive traits and some other dominant traits. So together, crossing over and random assortment can lead to 8 million different possibilities, and this is just within one gamete, not even including the fertilization process where gametes will join to create offspring. That process is called fertilization, and that is when we have a haploid ovum or egg combined with a haploid sperm. Both of these are gametes that have half the number of chromosomes needed. So they each have 23 chromosomes. When they combine, it creates a diploid zygote with 46 chromosomes. And this is where there is a lot more genetic variation, because the sperm and the ovum can be very wildly different, creating almost 70 trillion different combinations from 2 partners. This is why brothers and sisters are never exactly alike. Even paternal or fraternal twins are not exactly alike. The only case where you will see genetically identical siblings is if we have identical twins. Identical twins is different than fraternal twins. Fraternal twins means that we have a separate egg and a separate sperm that got fertilized within the same month. But an identical twin means we only had one sperm and one egg that created one zygote, and then very early on in its development, the zygote split in half, creating two different offspring that are genetically identical. We can see that process happening right here where we have an ovum, and you can see how much larger the egg is than the sperm. Now, genetically, the sperm is a haploid zygote, and the egg is also a haploid zygote. So why is the egg so much larger? Well, that is because spermatogenesis, or the formation of sperm cells, is very different than oogenesis, or the formation of an ovum. Spermatogenesis happens exactly how we just described. It goes through mitosis 1, and then it goes through mitosis 2. We end up with 4 genetically different gametes that then get formed into sperms. But in oogenesis, we go through meiosis 1 while you are still in gestation. So this happens within babies before they're even born. But if you've ever heard the term all your eggs in one basket, well, that is happening here. We're going to put most of the cytoplasm and most of the organelles all in one side, and the other one will not be viable. We call it a polar body, and it will not ever produce an ovum, but it kind of gets stuck to the secondary oocyte. It kind of gets stuck to it. In fact, you can kind of see one right here on this GIF right here. Then during ovulation, we will go through mitosis or meiosis 2, splitting again, creating another polar body, which can also get stuck to the ovum, which we can see over here. So that looks like a polar body, and this other one over here looks kind of like a polar body to me. And this happens every month in the woman's cycle, so long as she is fertile. So we end up with 1 ovum during oogenesis, and we end up with 4 sperm during spermatogenesis. Here we can see the process all together. Here we have prophase 1. We have the tetrads lined up, and crossing over can happen here in prophase 1. Then metaphase 1, everything is lined up on the metaphasic plate, and we can have half of the DNA going in one direction or half the other direction. This is called random or independent assortment. Anaphase 1 splits everything in half, and we end up with 2 regular genetically unique daughter cells. Then we go through prophase 2, metaphase 2, anaphase 2, and we end up with 4 genetically unique haploid gametes. In which stage do we have haploid genes? Only in stage number 8. In which stage do we have diploid genes? Well, we have diploid genes like right here. These are all diploid as they're being divided, but I would say number 5. And then when do we have a tetrad? Well, we have a tetrad right here in prophase 1. That is only because we duplicated the DNA in S phase. Let's watch a short video of this happening. Don't worry that there's no sound. We can just see the visual. So in prophase, we see the DNA condensing into chromosomes. They will line up in the tetrad. This is also where crossing over might happen. Then in metaphase, they line up at the equatorial plate, randomly aligned to either pole. So we don't really know whether the DNA is going to go to the top or the bottom. Now in metaphase, we're going to play that tug of war, and it will pull apart to either pole. during anaphase 1. Now we still have 2 copies of all the DNA. So we call these new cells, the daughter cells, will be diploid, but they will be genetically unique diploid cells. So here we have telophase, and we have a diploid daughter cell here and a diploid daughter cell here. We're going to go through all of those phases again. Prophase 2, metaphase 2, and anaphase 2, and telophase 2. We end up with 4 genetically unique haploid gametes. Don't forget to print your chapter laboratory write-up and complete your lab quiz, OpenNote lab quiz. Don't forget to complete your chapter quiz in lecture, which is closed notes. And if you would like extra credit or extra help, you can do the chapter homework.